Okay, so we are going to start the third session of this meeting, where we're going to focus on the social and affective behavior and cognition. And I invite the first speaker, Birgit Derntl from the University of Tübingen. So just a short introduction. She's a psychologist and psychotherapist by training, and she's a full professor for innovative neuroimaging at the Department of Psychiatry and Psychotherapy. She's going to talk about the interactions between emotion and cognition. And the title is Mapping Regulation Process in the Female and Male Brain, which gives me the opportunity to say how happy I am to chair a session in which women are so well represented. So I'll give her the floor, and thank you for your talk.
Thank you, Terry. So that was the title that I came up with when Mark asked me whether I want to contribute to the symposium. Of course, I want to contribute. So thanks for asking and for giving me this opportunity. But the title, I'm not so sure. When I started preparing for the presentation a few days ago, I thought, oh my God, what did I think? So I came up with other titles. I dismissed them. So who needs a title when you can have such a wonderful building? And this is the Department of Psychiatry and Psychotherapy here in Tübingen. And it would be wonderful to actually welcome you here, as this is the place where I'm working. I'm not sitting, I'm in home office right now, but this is where I would sit if I would be in the office. And it's very nice. And here also our patients treated some of our patients are also here. So this is also part of the Tübingen Center for Mental Health. Now, it's more about today, I thought it's more about presenting the lab to you and what are we doing and what is our research focus. So I think I start with who are the participants in our research lab. And now we're coming to humans. So this is human neuroimaging now. And I think 1/4 of our participants is males and transgender individuals, and the vast majority of our participants are females. Now, how come? Because in the lab, we are interested in hormonal transition periods, including the menstrual cycle and hormonal contraception, pregnancy and the postpartum period, but also the perimenopausal phase. So that's why we have this focus on females and focus on women. But I give you 3 examples from ongoing studies in the lab. And I just start with one where we are also interested in sex differences. And that is the domain of stress. Now, stress is not really something that I need to introduce to you. But of course, working in the psychiatry, being a psychologist, psychotherapist, stress is something that is related to mental health. And it's a common risk factor for mental disorders. So it's something that we need to understand and investigate. And we also need to understand the mechanisms behind stress and stress reactivity. Now, how do we introduce stress in the neuroimaging environment is always a good question. And there are some methods available that you can use in the MRI scanner. So that's the method that we are heavily using, functional magnetic resonance imaging. Here, and together with colleagues from Aachen University in a DFG-funded project, we are using the scan stress. And the scan stress is just one of a handful of stress induction methods that have been used in the scanner up to now. And we've done them all in the lab. But for now, I introduced the scan stress to you, which is similar to the others in terms of having a cognitive challenge. So you have to do mental rotations and mental arithmetics here in this task. And the social evaluation component, very important for stress induction. There are 2 people watching you while you are lying in the scanner, while you are performing the task. These 2 are watching you closely. And if you do an error, it's not only shown to you via this red information, they are also pressing a heavy red buzzer. And so you can immediately see that they are not satisfied with the performance that you are doing here. So classical stress induction, not very nice, but we need to investigate that because that's what we are going through. So this is work by a postdoc in the lab, Hannes Noack, who, together with colleagues from Aachen, collected a sample of 140 participants. As you can see here, females and males. And this is the first parameter that, of course, you would be interested. It's cortisol. So how does the cortisol react to the stress induction? And as you can see here, we do have several responders. So male responders in blue, and the female responders, meaning that they show a cortisol increase in red. But there are also the dotted lines. So we also have non-responders, cortisol non-responders, and that happens. We don't know why, but it happens. When we then look into the subjective stress, there, of course, you also see sex differences. So females report to be more stressed than males. That has been reported by other groups before. But again, if you differentiate by a cortisol responsiveness, you see that the female cortisol non-responders show a similar subjective stress response than the male cortisol responders. So it's something that there are different levels. It's something that we ask our participants, but we have to integrate that data. And of course, if we go to the next level, to the brain, then you have sex differences here. So the males show a stronger neural activation compared to the females. And this is irrespective of being a cortisol responder or non-responder. So integrating all of this data is something that we do and that we are interested in. And here we ran a regression analysis, taking the factors of sex, of cortisol, of subjective stress. We also measured heart rate. And we all took this and wanted to predict the neural activation. And the only thing that worked was subjective stress in men for the hippocampal, parahippocampal region. Now that has been shown by others as well, as the hippocampal region is crucial for stress reactivity, and the hippocampal connectivity can actually predict feelings later on. There's one paper showing this, but still what I wanted to show you with this example is that we have to look at different levels and that we have to integrate these levels to better understand the stress reactivity, because this is a complex phenomenon, as most of human behavior is complex. So we have to address this also by looking into different levels and then integrating them later on. Now, we have several research questions that we want to follow up or that we are currently addressing in the lab. Hannes, for example, is also looking into the consequences of stress on cognition. So what happens if you've stressed them, the females and males, and now they have to perform a working memory task in the scanner? How do the cortisol responders respond? How is subjective stress predicting the cognitive performance later on? This is what Hannes is currently analyzing and working on. Together with Vanessa Nirajka, the head of our molecular psychiatry group in the Department of Psychiatry, we are looking into the impact of genotype and epigenotype on this data. Lydia Kogler, a postdoc in my lab, is the person to go to if you're interested in transferring data from the lab to everyday life, but also when you're interested in training and interventions, because she's just done a study where she used a training on positive self-instruction and tried to see whether this is actually reducing the stress reactivity in females. It did. And now she's finally analyzing the data and also writing this up. So how do we cope with stress? How do we regulate stress? That would be topics that Lydia is interested in. And also, of course, the translation from healthy participants to the clinical populations, here depression, but we've also done research in schizophrenia patients, for example. And I mentioned the females being the majority of our participants. And so Zoe Bürger, a doctoral researcher in the lab, is interested in how hormonal profiles and in particular hormonal contraception is affecting the stress response. And she is interested in intrauterine devices. And this is also, as this symposium is about getting to know each other and potential collaborations, this is also a call out to all of you who have neuroimaging data and probably also have data, structural resting state data on females having intrauterine devices. We don't have any publication on this up to now, so we don't know what intrauterine devices do to the female brain. And it would be wonderful if someone of you has data to share that and to collaborate in this regard. Now let me come to the females and the menstrual cycle and hormonal contraception. And here I want to focus now on empathy, another human complex phenomenon that we are interested in the lab. And empathy is, there are several definitions. It kind of always depends on who you're asking. They come up with different definitions, but here I think this is a very general one to say that it's the ability to recognize others' affective states and share the emotional experience, but always being aware of the self-other distinction. And I think we've agreed in the field now on two components of empathy, the one being the perspective taking, the mentalizing aspects, or more the cognitive empathy, and the other being affect sharing and affective experience, and that's more the affective empathy part of it. And we've done several studies on empathy and also empathy in schizophrenia. That was one of the focuses of the lab in the past. But we never were satisfied with the tasks that we used. So it was time to develop a new task. And together with Dirk Wildgruber here from the Department of Psychiatry, a colleague, if you're interested in prosody, if you're interested in auditory stimuli, in laughter and humor, then Dirk would be someone to go to. Together we developed a new task called the, of course, Tübingen empathy task. And what we tried to do here is to better differentiate between the cognitive and affective component. And it works like that you imagine a situation and we ask you, if you are in this situation, how would you feel? That is the self component. And then, of course, as empathy is about others and the self-other distinction, we have two other conditions, one being the friend or the liked person. So if you think about your friend in this situation, for example, she goes on a concert of her favorite artist, in this situation, she feels, and that is perspective taking. So this is cognitive empathy. How does another person feel in a certain situation? Then they give us a response, a rating. And then we ask, and how do you feel knowing that your friend is in this situation? So is in the concert, for example. So what does that do to you? And that's the affective experience. And we not only have the friend, we also wanted to see how does that change if you take a disliked person, an enemy. But dislike is something, we don't have that many enemies, actually. We rather have disliked persons. So this is another condition that we used, and we tried to separate between this affective and cognitive component. And Ann-Christine Kimmich, a doctoral researcher in my group, is applied the Tübingen empathy task in the neuroimaging setting. And she, of course, was then interested in how does the natural cycle or the cycle phase and oral contraceptives actually affect empathy. And this is also a DFG funded project that we are running. And when Ann-Christine used this Tübingen empathy task, she recruited a big sample of females because she's interested in oral contraceptives. So we had to have these females taking oral contraceptives, but also natural cycle phase. So she had females in this FNC group, that's the follicular natural cycle. So it's the early follicular phase. It's the first days of the menstrual cycle when estradiol and progesterone, the two sex hormones, are low. And she also had women during the ovulation phase. So that's the middle of the natural cycle when estradiol is actually very high and the females are most fertile. And these two natural cycle phases were contrasted with the oral contraceptive taking females. Now, if you look at the behavioral results, we see, or Ann-Christine demonstrates that the oral contraceptive taking females show reduced cognitive empathy, but also reduced affective empathy. And it doesn't matter if it's the friend, the enemy, or the self, it's reduced. When we turn to the brain, we see a similar pattern in that the oral contraceptive taking females show less neural activation compared particularly to the ovulation females, so the females that are fertile with the high estradiol levels. And when we then try to connect the brain and the behavior, we see a significant correlation, meaning that the higher the cognitive ratings, the stronger the activation in the brain. But also for the first time, Ann-Christine analyzed the impact of the synthetic hormones, because when the females are taking oral contraceptives, they suppress the endogenous hormones and have the synthetic hormones. And now we, for the first time, show that there's also correlation. So the higher the synthetic hormones are, the higher the synthetic estradiol, the less brain activation we are seeing. So in summary, what her data indicates is that oral contraceptives do influence empathy. We didn't find much of a difference between the natural cycle phases, but the oral contraceptives apparently influence empathy. And of course, there are several questions that we want to address here and that we want to take also in future studies. It's of course about the immediate and the long-term effects of contraception on brain architecture and behavior. And Christine has longitudinal data in these females, and she also has a group of females who start taking oral contraceptives, and we've measured them before they started, and we measured them while they take oral contraceptives. And she also has a group of females who were measured while they were still taking oral contraceptives, then finished, and we measured them when they are not on oral contraceptives. So we can actually see on what it does. Are these changes reversible, for example, if you stop taking oral contraceptives? Oral contraceptives and what do they do to sexual appetites or psychosexual health? Another question that we want to address. For this, we have developed an erotic approach avoidance task. Of course, that was something that we all contributed to, looking at the pictures. And this is also something, I'm not talking about this right now, but it's something that we are, of course, interested in. And can we classify who is naturally psyching or taking oral contraceptives based on brain parameters is also something that Anne-Christine is, of course, curious about. And as always, we are interested in the translation into the clinics. Empathy, we've done lots of research in schizophrenia. So of course, we are interested in also using our Tübingen empathy task in these clinical populations, such as schizophrenia patients or individuals with autism spectrum disorder. And my third example is now about pregnancy and the postpartum phase, but I stick to pregnancy. And I want to talk about emotion regulation. So another complex ability of us humans. But first, let me address pregnancy. I think pregnancy is something, when you talk about the neuroimaging setting, we don't have that many samples of pregnant data sets or pregnant brain data sets. So this was something that we started with our colleagues from Uppsala University in Sweden, and which was also funded by the Center for Integrative Neurosciences, which was presented to you by Cornelius Schwarz today in the morning. And here we jointly supervised 4 doctoral researchers together with the Swedish colleagues. And one of them, Elisa Rehbein, in my group, she acquired the biggest neuroimaging sample of first-time healthy pregnant women. It was 13 at the time we published the data. Right now we have 15, still not that much. We're still measuring, but nevertheless, just to let you know that what we see right now, or what Elisa reported in one of her papers, is that there are structural changes as early as the 2nd trimester in the brain. And this here is the putamen, where we see the earliest changes in volume in the pregnant women. As you can imagine, Corona did not help in recruiting first-time pregnant women. So this is something where we really are working on and try to collaborate with other centers to get bigger samples, of course. And also another colleague of mine that I want to introduce here is Hubert Preissl, because he is part of this pregnancy and the brain mini group, and he is the one who's doing the fetal measurements. So while we were measuring the mothers and we're interested in the maternal brain or in the pregnant brain, Hubert is the one who is investigating the fetal activation, and now we can then see how pregnancy development and also fetal development and later offspring development are actually associated and working together. But coming back to our pregnant sample, Melina Gralo, a doctoral researcher in my group, is interested in emotion regulation in pregnant women. Now, that, of course, emotion regulation contributes to mental health also in pregnancy and the postpartum period. There have been some questionnaire data available. It's not much of research on emotion regulation in pregnancy. And if we give our pregnant women a questionnaire on self-reported emotional regulation use, we see that they use reappraisal, a cognitive emotional regulation strategy, less often than naturally cycling women. And here, the labels may be a little bit confusing because here you see E2 and PC. What E2 means is that this is a naturally cycling group where we administered estradiol to push the estradiol levels up into the levels of ovulation within 24 hours. And the PC group is the placebo group. So they took the placebo pill. They are still in the early follicular phase. They still have this low estradiol levels. And now they performed this fMRI emotion regulation task. It was a very simple task, not too long, and they just had to view or down regulate while watching a negative stimuli. And what you see here from the behavioral performance is that the pregnant women can regulate negative emotions. At least when instructed to do so, they down regulate and they can do so. So there's no group difference. And also, tentative and very preliminary data from the brain activation, but that's the first fMRI study in pregnant women, showing that we don't see a group effect. Although it looks like there's something going on, this is not statistically significant, probably due to the sample size, of course, with only 13 pregnant women. That is very low. But still, it shows us that there is something going on and we have to further look into that. We are still recruiting. We're hoping to reach 20 at least at some time and then present more elaborate analysis. But emotion regulation as a transdiagnostic factor, I think, is something that we are aware of. There's lots of research on emotional recognition and dysregulation of emotions in patients with mental disorders, most of the time focusing on mood disorders or anxiety disorders. And what is interesting and also important is to again look at the different levels, because if you look at the studies here and you look at the self-report, so if you ask the patients, can you regulate your emotions, and they fill out a questionnaire, there's no group difference. So the patients report, yes, I can do that. But if you then do a behavioral task, or if you even go to the scanner and do an fMRI measurement, the studies report group differences. So meaning that these are complex phenomena that we really have to address at different levels, and we have to see how they also interplay with each other to better understand what's going on. And this is also now my discussion, emotion cognition interaction. Although I wasn't too happy with the title, I think, yes, you hopefully saw that many abilities that we are interested in have this emotion cognition interaction, and they may also be seen as transdiagnostic factors. So I think stress and emotion regulation is probably not that complicated to see that. For empathy, we probably have to discuss that. In the lab, we want to have an assessment on several levels. So we always have self-report, but behavior, physiology, hormones, and neural activation are also collected, and then we have to integrate the data. We are particularly interested in women's mental health. I think I outlined that, with a focus on hormonal transition periods. Now, why are we so keen about hormonal transition? It's because every hormonal transition period is associated with a higher vulnerability for mental disorders in women. And that's what we want to investigate and what we want to better understand. Because there's a gender gap in prevalence rates. And these are the prevalence rates for common mental disorders from 2019. Sorry, it's for Germany and Sweden. That's the partners of this Pregnancy and the Brain consortium. But I think it would be very similar if I take the French or the Italian data. And you see that women are at higher risk for developing a mental disorder, a common mental disorder. And this starts with puberty and then it stays up. And we know this hormonal transition periods have this higher vulnerability. So we think that better understanding transdiagnostic factors may improve the prevention, detection, and then also the treatment of mental disorders. That's why we are so keen about better understanding these complex phenomena and trying to really break them down on the separate levels and then integrate them, of course, back again. And with that, I'd like to end. I'd like to thank you, but of course, I'd like to thank the group. I didn't present all the studies that we are running, and it's just amazing to work with this group and to have these wonderful doctoral researchers and postdocs in the lab, and also the students contributing to that. We have a lot of collaborators we are working with, and hopefully there will also be collaborations coming up from the CIVIS network, and of course the funding agencies that support us in running these studies. So thanks very much. And now I'm curious about your questions.
Can you hear me? Thank you. Thank you, Birgit. So my name is Rochelle Ackley. I'll be helping with some of the questions as well. A really, really interesting talk. Thank you very much. It's a very important area on women's mental health. We've already got one question. I don't know if you can see it in the Q&A, but it's from Laura Donnelly, who's asking out of curiosity, have you observed the effects of synthetic hormones on transgender individuals' emotional regulation?
So thanks for asking that, Laura, and thanks for posing the question, Rochelle. So in the transgender study, we are indeed looking into the effects of hormonal therapy in the individuals. So we measure them before they start with the hormonal therapy, and we measure them while they are 6 months under hormonal therapy. We are in the middle of recruiting. So this is an ongoing study. I don't have any results for you now on that. And we, together with a colleague from the Pharmaceutical Institute here in Tübingen, we've developed a method to actually analyze the synthetic hormones. So we could analyze this and we could then see what's happening there. But we just did that for the oral contraceptives, and we will do that in the future for several other projects whenever there are synthetic hormones given to participants. So yes, we'll do that in the future, but right now I can't answer this question. And we're not doing an emotion regulation task in the transgender study. This is more about gender and gender perception and self-perception. So gender identity that we are asking in our tasks here. It's not about emotion regulation.
Okay, thank you. Do we have any other questions coming in, either in the chat or the Q&A? Laura says thank you. Thierry, you have a question?
Yeah, thank you. Thank you for your presentation. I just had one thing that surprised me a little bit. I just wanted to ask, in the one fMRI study you presented, there were some cortical activations related to the cycle phase, if I remember correctly. I was surprised, and I don't know if it's just because you didn't have the time to mention it or otherwise, if there isn't any subcortical delay to actually deal with the effects that differences in circulating hormones could have on the cortical processing. So do you have any subcortical targets for the effects of hormones on the women's brain?
So yeah, of course. So there are several subcortical areas that we would be interested in, but they are hard to actually get. It's the hypothalamus, of course, that we would be interested. It's, of course, the striatum is something that we are also interested in. This we can measure pretty fine. And it's also the tegmental area that we would be interested, because whenever it comes to emotions, whenever it comes to any social aspect, I think this is also a region that is crucial. Again, hard to measure. So this is something that we didn't optimize our imaging protocols for right now, but the striatum is something that we can cover and that we can also measure.
Thank you.
Sure.
No, it's really interesting. Because I have a quick question. It's more of a general question because it's something that I've observed in my own data, but it's about responders and non-responders. So you see responders to stress and obviously non-responders. I was wondering if you think this is more stress specific or not, because I've seen this in tasks which are not particularly stressful, where we have responders and non-responders when we measure physiological mechanisms like electrodermal activity and the sympathetic nervous system. So I was wondering if you can comment your thoughts about this.
Mm-hmm. I think, Rochelle, that is something that we need to investigate much more and need to better understand what's happening here. We had one neurofeedback study where we dealt with non-responders and nobody knew why they are non-responders. So this was really puzzling for us. And I think for the cortisol, I do have some, I guess we have some theories why they are non-responders and so forth. But is that a general aspect to it? Is that something that we can translate then to other measures as well? I'm not sure about that, but I think it would be worthwhile to spend much more time on the non-responders than to only report about the responders that show the profile that you were expecting. So I'm absolutely with you. I think it's puzzling, it's interesting, but I don't have an answer why that is the core, and if there's a generalization to that.
No, thank you. That's really interesting for me as well. I think maybe one final question from Santiago.
Thank you, Birgit, for this nice talk. I'm far from being a specialist in the area, but I dare to ask a very naive question. How these processes affect decision-making processes?
Mm-hmm. Yeah. That's I think decision-making is here in every aspect. I mean, they have to perform a decision more or less in every task that we are doing. We have actually one doctoral researcher of mine is interested, Esther Ambrase. She is interested in moral decision-making and how moral decision-making is affected by sex, but also by hormones. And here we do see sex differences. So for the moral decision-making domain, we do see sex differences, of course. And we are trying to get at the point how that is related to gender, for example, because when I talk about sex, it's much about the biology, it's about the menstrual cycle, etc. But of course, there's a gender to it as well. And this is also part of what we are interested in. And so we're trying to integrate that. And that is all I can say about decision. I'm aware of a vast literature on decision-making and sex differences, on decision-making and menstrual cycle. It's not that much, as always with the menstrual cycle. And I think on oral contraceptives, there's not that much on decision-making too.
What about stress?
Stress. Oh, in terms of stress and the consequences of stress on cognition, I think the best investigated aspects are inhibition and working memory. So there's a meta-analysis on this, and it showed us from behavioral studies, and it shows us that working memory seems to be impacted by stress in terms of that it gets you get poorer in your working memory after stress induction, but inhibition is not that much influenced by stress. At least that was the results by the meta-analysis. And so we decided to look into working memory based on the results of this meta-analysis to also find out how do the responders, non-responders, how do females, males, and you have it because there are several factors to it, how do they then show later on working memory performance? So is that differently affected based on whether you've responded to the stress induction, whether you are a female or a male, and of course the genotype, epigenotype is also something.
Thank you very much.
Sure.
Thank you, Birgit. I think I'll introduce our next speaker now, who's the local speaker, Hélène Grosbras, who joined Aix-Marseille University in 2015. She's working in the Laboratoire de Neurosciences Cognitives, and in collaboration with the fMRI Center, her group investigates how the adolescent brain develops in relation to changes in social cognition. So I will give the floor to Hélène now. Thank you.
Thanks for the introduction, and thanks Santiago also for inviting me to this exciting symposium. I'm very honored and pleased to be able to share some of our work to so many researchers from the different universities. I'll start with a short introduction of the general topic of the research we carry out here in the laboratory, in my group in the Laboratoire de Neurosciences Cognitives. So the way we plan and execute many of our everyday actions is influenced by our social surrounding. And conversely, our actions will influence other people and the way we perceive them. So in my group, we're interested in understanding this bidirectional reciprocal interaction between action control, high-level action control, and social perception, and in establishing to what extent it reflects a special processing of social stimuli as compared to other objects or events. So to this end, we look at that in the lab with laboratory tasks, and also use mainly functional magnetic resonance imaging to look at the related brain activity. And a large part of our work examines this question in adolescence. Indeed, adolescence is a time when both social processing and executive control and the underlying brain networks are both still undergoing important maturation, albeit with different time courses as a function of the different functions. So we might expect a special modeling of the interaction between action control and social perception at this period of life, which is also, as we've just seen in the previous presentation, a period of high vulnerability to mental disorders that will affect both cognitive control and social perception. So today I'll present you two representative studies. In the first one, we looked at action toward the social stimulus, and the second one at the observation of action toward the social stimulus, comparing adolescents to children and adult participants. The first study starts from the hypothesis that given the asynchronous maturation of different aspects of cognitive control and social cognition, we might expect that the high-level mechanisms involved in action control will be impacted by social context differently as a function of the period of life during development. And that this interaction between social and cognitive processing at the cerebral level will also vary with age. So in this study, to address this question, we used a classical task employed to assess executive control, in that case, inhibitory control. That's the antipsychotic task. So in this task, you first see a fixation cross, which is after a period of time is replaced by a cue, followed by a blank screen just before a target is flashed in the periphery. So if the cue had been green, the task is to perform the reflexive action, that is to look toward the target. But if the cue had been red, then the instruction is to inhibit this prepotent response and instead to look away from the target in its mirror location. So antipsychotics involve a high level of control as they require to inhibit the reflexive action and to reprogram an appropriate voluntary action. We use this task with different kinds of targets, either social target, so in our case, faces, non-social objects, but still meaningful objects, in our case, cars, or meaningless noise patterns. And first, in a behavioral study, we tested 139 participants between the age of 8 and 22. What you see here is the average error rate. That is the number of times participants looked at the target when they were supposed to inhibit this reflexive movement as a function of the stimulus type, so either face, cars, or noise patterns. And I hope you can see my pointer. And what you can first see as a function of age, so from children to young adults, there is a decrease in the number of errors reflecting an important improvement in inhibitory control. As for the effect of stimulus, so first, if you look at the young adult, you see that they make significantly more error for faces than for cars or noise patterns, meaning that it's more difficult to inhibit an eye movement to a face than to any other stimuli, reflecting the special effect of social stimulus of the face, in this case, to the inhibitory control. For adolescents, we see also that faces induce more errors than cars, but not differently to noise pattern. For the early adolescents just entering puberty, there's no stimulus effect whatsoever. And for these children, they have more difficulty in the task, they make more errors for both face or cars, meaning any relevant or significant stimulus as compared to noise patterns. So what this shows that the specific privileged processing of faces with regards to executive control seems to appear at the end of adolescence. So we looked at this in the scanner to look at the related brain activity, and that was the work of Alia Afyouni, a PhD student in the lab. So in that case, we took only face and car stimuli, and we scanned children, adolescents, and adults. So first, if we look at the brain activity involved in inhibitory control by contrasting anti-saccades to pro-saccades trials, we see that in children, adolescents, and adults, more or less the same regions are activated. And if we summarize this activity in the right side of the slide, we see that in all of these regions, so it's significant, they are all significantly involved in inhibitory control in all age groups, but the strength of the activity increases with age between childhood and adolescence and between adolescence and adulthood. For the stimulus effect, comparing trials with the face, trials with the cars, we see also that regions that are expected to be more involved in faces than objects or than cars are activated in all age groups, but with some developmental effects. So in the fusiform face area, region specific to processing of faces, we see an increase in activity from childhood to adolescence, but no difference between adolescents and adults. And in the amygdala, there was no effect of age when we started with between children, adolescents, and adults. So now the most interesting question was the interaction between the task and stimulus. And we asked, where are they part of the brain when there will be higher activity in this inhibitory controller network, or for the trials with a face as compared to a car. So where people would more activity for anti-saccades to face than to cars. So as expected for children, we didn't see any activity anywhere, any interaction anywhere in the brain. But for the adolescents and adults, we could see that there were regions where this activity related to anti-saccade was, compared to pro-saccade, was higher for faces, but it was different in adolescents and adults, differently located in adolescents and adults. In adolescents, there was located in occipitotemporal region and superior temporal region, postcentral gyrus, that's regions that are often associated with social processing. Whether in adults, this interaction took place in a prefrontal region, in superior frontal gyrus, a region that is involved in high-level control or spatial orienting. So a summary from this study, we see that we could evidence the improved inhibitory control with age, both in behavior and in brain activity. And we saw that social stimulus affects inhibitory control differently in children, adolescents, and adults, with a kind of special effect of faces appearing at the end of adolescence and relying on different brain activity in adolescence as compared to adults. In the second study I'm going to present, we looked at a different aspect of interaction between action control and social perception. Because indeed, actions of other people are in themselves a social stimulus. And when we observe someone else moving, someone else action, there is a set of brain regions that is consistently engaged that we call the action observation network, involving frontoparietal regions as well as occipitotemporal regions. And remarkably, there is a strong overlap between these regions, so involved in social processing of actions, in action as a social stimulus, and regions that are involved in planning and executing those actions. And also of interest that the activity in this region during action observation is motivated by the nature and content of the action, with stronger activity for object-directed actions, and also, of most interest to us, a modulation and with stronger activity when the actions have a social dimension, that is, they involve another person. So in this study, we asked this following question. We asked whether this representation of action categories in the action observation network will change during adolescence. And we expected a more important differences between adolescents and adults in the social dimension, given the ongoing development in the underlying networks at this stage. So for this experiment, we built a set of stimuli consisting in action videos in a 2x2 factorial designs. So the videos could be either social, that is the action pictured in the video involved the other person. So there was always one actor and one person not moving, or non-social. Or they could be what we call transitive, that is they involved an object or not. So an example of a social transitive action would be, for example, when the actor gives or takes an object from the other person. An example of a social intransitive action would be when the actor makes a gesture that is significant to the other person. An example of a non-social transitive action would be manipulating the object, but without any relevance to the other person. And an example of a non-social intransitive action would be a gesture that has no relevance to the object nor the other person whatsoever. So we showed those videos, or a large number of those videos with different actors, different objects, to a sample of adolescents and adults in the scanner, and we looked at the brain activity. And that was the work of Mathieu Lesour, a postdoc in the lab. So I'll just present a brief snapshot of the results. First, when we looked at the effect of sociality, that is comparing actions with the social dimension versus non-social actions, we replicated previous results showing that areas of the action observation network are more involved in social than non-social action. And when we looked at the transitive factor, also we replicated previous finding of higher activity in some regions for transitive compared to non-object directed actions. But with this analysis, we couldn't see any difference between adolescents and adults, meaning that the modulation of activity in these regions by the social dimensions or the object dimension was similar in adults and adolescents. But we decided to investigate this data a bit further using another approach, which is instead of looking at the difference in fMRI signal between condition voxel by voxel and then averaging, we'll take regions, those regions of interest where there is the most modulation by those two dimensions, object and social, and take the information given by the ensemble of voxels, the set by all the voxels. So how we do, we define those regions of interest, and for each participant and each voxel and each trials, for each trial, we will extract the pattern or put the activity in each voxel in a vector and extract this pattern of activity. And we do that for all trial. And then we can use machine learning to try to make sense of those patterns. So concretely what we do, we take a part of the data and we train a classifier, so for example, a support vector machine, to associate the pattern of activity across all the voxels of this given region, to associate the pattern of activity with the nature of the stimuli, for example, social and non-social. And then we take this classifier and we test whether with the classifier we can decode the pattern of activity recording in the other part of the data. And if the reasoning is that if the classifier is able to above chance successfully guess the stimulus based on the pattern of fMRI response activity, that will mean that the classifier has learned some information relevant to distinguishing social from non-social stimuli, and thus that in the regions that we're investigating, there is a representation of the social dimension. And so we can do that within the orthogonal dimensions, that is, for example, training to distinguish social from non-social action within all the transitive actions and test it within the same dimension, to testing the social transitive to non-social transitive. And separately train another classifier to dissociate, to associate social or non-social actions that are intransitive, and turn them on intransitive actions and average. And if we see we see significantly above chance decoding, that will tell us that the region where we're looking has a general representation of sociality. But also we can do that across the orthogonal dimensions. That is, for example, trying to dissociate social from non-social within the set of transitive actions, and see whether this classifier would be able to decode social from non-social actions that are intransitive. And if this is done above chance, that would mean that there is a more abstract representation of the social dimension in this region. So what Karamazov would call a conceptual representation of sociality. So this analysis gives us a more fine-grained information about what's going on with respect to the representation of those two dimensions. So we run that on those three regions. And what you see here is performance or average performance across sessions and participants of the classifier in the lateral temporal occipital cortex, parietal cortex, and ventral premotor cortex in light color for adolescents and in darker color for adults. And first we can see that in all regions in adults and adolescents, there is significant decoding of the social dimensions, meaning that there is a representation of the social dimension in all regions, but it's strong, like the performance of the classifier is stronger in adults and in adolescents in the right lateral occipitotemporal cortex and bilateral parietal and premotor regions, meaning that, so that's interpreted as the fact that maybe the representation of the social dimensions in adolescent is not as stable or as reliable as it is in adult, and that it's still maturing. We see the same in those three regions for the transitive dimensions, and with the same difference between adolescent and adult. Now, when we look at the across category decoding, so looking at a more abstract representation of these different action categories, we see that only in the lateral temporal occipital cortex and ventral premotor cortex, there is this abstract representation, but not in the parietal regions. And what's interesting here is that in the regions that's more sensitive to these two manipulations of the content of the actions, so the LOTC, there is a difference between adolescents and adults bilaterally in the left hemisphere and right hemisphere, but only for the social actions and not for the social dimensions and not for the transitive dimensions. So highlighting a weaker abstract representation of sociality in the adolescent lateral temporal occipital cortex. So in summary of this study, we've seen that the adolescent action observation network is similar to the adults one and sensitive to the nature of the action and shows a fine-grained representation of the action category. But nevertheless, there is still some maturation going on as the decoding accuracy was lower in adolescents for both the social and the transitive dimensions and the object-related dimensions. And we could see a weaker abstract representation, conceptual representation of the social dimension in the lateral occipitotemporal cortex. So to conclude, like kind of summary of those two studies, they show that the processing of social stimuli, so being faces or actions, that shows only subtle differences between adolescents and adults. But in first study, we saw that in the context of action control, the difference between social and non-social stimuli was different as a function of age, and the privileged processing of the social stimuli started to emerge only at the end of adolescence. And the brain representation of the execution, as in the first study, or observation of the socially directed actions appears a bit less robust in adolescents and still in a period of maturation. So we're still pursuing this work, trying to relate those results to baseline functional organizations, that is functional cognitivity at rest, and to various aspects of behavior. And to finish, I would thank all my collaborators and the students and postdocs in the lab, as well as the engineer of the fMRI Center. And I thank you for your attention.
Okay, thank you very much, Marianne. That's a very interesting talk on the changing brain, especially in adolescents. It's clear there's a lot of changes going on there. If we have any questions, please feel free to post them in the Q&A part or in the chat. But I can start. I have a question about the separation of, you have, I think when it's about 14 years old, the person has a very changing brain, and they have a social component, and they also have a decrease in the errors as well, so the inhibition. And I was wondering to what extent are these processes separate, or if they're always combined?
So you mean the maturation of the cognitive control compared to social processing? No, they're not combined all the time, and it really depends of the level of the functions that we're targeting. So different executive functions will mature not exactly at the same rate, and the same way different aspects of social cognition will mature at the same rate and at slightly different rates. So it's really hard to look at the relation between the two, because the two are changing at the same time, and at the same times, but on different timelines. And also they are kind of like, we can't really look at with this kind of focused fMRI studies, these inter-individual differences. So what we try to do is to have homogeneous group in terms of, as you say, like pubertal maturation. That's what we looked at as in relation to the previous talk, and also like socioeconomical status and so forth. But still, it's something we cannot really look at, because, as you say, there are also many social changes at this age. So yeah, it's really hard to make the part of all those things. But so with our kind of homogeneous group, we can see that for some functions, like the inhibitory function and the face processing, or basic passive perception of faces, they are not developing at the same rate. Yeah.
Well, that's really interesting. A lot of variability there, which can make things obviously difficult to analyze. Mm-hmm. Thierry, you have a question?
Yeah, thank you. It's kind of a similar question that you had, but mine is more about the adolescent versus adults in the interaction term of the fMRI experiment. I was very surprised, and I actually wondered if it was by design or if it was an actual result, that you had nothing in common when you did the interaction between the anti-saccade and the face processing in the adolescent and the adults. If I recall correctly, the adolescents identified regions of social, and the adults was kind of inhibitory control. But you didn't mention any commonality, as if it was like a big change between these two age groups.
Yeah, in the interaction, yeah. And like when you take the look at the task effect or the stimulus effect, there are lots of overlapping effects. But in the interaction, there's no overlap whatsoever. It's completely different networks that are sensitive to this interaction.
Okay, and do you have any interpretation, or it's just maybe?
I think that it doesn't mean the same thing for them, and just behaviorally also that was a bit different. In the fMRI, we looked only at car and faces, where they show more or less the same behavioral effect, and we didn't have those abstract stimuli. No, my interpretation is that they solve the task, or they are sensitive to the social stimulus, but by different mechanisms.
Okay, thank you.
Okay, are there any other questions? I'm gonna just ask one, maybe last one, a quick one about, you said about the representation in the adolescent brain is less robust. And I was wondering, does that mean it's more variable? And can you quantify the variability here?
It's a bit more variable. So we looked, I didn't present data. It's a bit more variable in when we do individual statistics in terms of how many adolescents will show like a significant effect at the individual level. So it's a bit less. In adults, it's almost all of them. It's significant even at the individual levels, whereas the adolescents are few. So there is some inter-individual variability. And there is also some inter-individual variability that we didn't quantify here. I wouldn't know how to do that with like the multivariate pattern analysis. With the univariate analysis, we could look at just the standard deviation, but we haven't really done that. But it's kind of common in developmental studies that you've got increase in those two variabilities. And what I didn't present also in terms of the inter-individual variability is that in the right hemisphere, when we try to look at multivariate MVPA result as a function of age, that was linearly correlated quite significantly actually. But yeah.
Okay, thank you. Thierry, should we move on to the next speaker?
Yes, I'm gonna introduce the last speaker of the session, Valeria Carola. She's a researcher at the Sapienza University in the Department of Dynamic, Clinical and Health Studies, as well as a clinical psychologist at the Sant'Andrea Hospital in Rome. So her work combines preclinical research with clinical studies in order to identify biomarkers implicated in the translation of early life social stress into behavioral and psychological alterations. So I give her the floor to Valeria Carola.
Yeah, first of all, I want to thank the organizer for inviting me to present my work today here. It's really a great pleasure, and I guess that's really a good opportunity for all of us to meet each other. And today I'm presenting one of my work. I was just to introduce myself. I'm a psychologist, and I work both on preclinical neural models and in a human clinical setting. And today I'm presenting a clinical study. The title of this study is Multidimensional Assessment of Psychopathological Risk in Young Adults Who Experience Emotional and Relational Trauma During Childhood. First of all, I want to mention that my group is mainly focused on the investigation of the changes that are associated with experiencing an adverse childhood, and these changes, how these changes are related with the expression of psychopathology later on. We work on this topic both on neural models and in clinical settings and clinical studies. And as many of you know, there is a huge literature about the connection between adverse childhood experiences and psychological health issue and also changes in physical health and medical health. So we all know that being exposed to adversity in early childhood has a huge impact on individual and on really broad different aspects. The main aim of our group of my group is to investigate how such experience is translated later on in psychopathology, in which type of biomarkers are connected to this translation. So in order to have the possibility also to, predict the susceptibility to psychopathology after the birth childhood, or having the possibility to do some personalized treatment related to the type of experience that we had in the past, and also related to the genotype of each of us. Mainly one of the main aspects that is really relevant for us is to investigate also not only the relevance of the complex trauma that we experience in the childhood on the later on outcomes, but also the relevant.
Excuse me, Valeria.
Yeah.
Can I interrupt you a second? We're having a little issue with your microphone. I don't know if there's something we can solve easily. It's kind of resonating. So it gives kind of a background noise and make it a little bit harder to actually get you.
Okay, can you hear me a bit more better? Can you hear me better now?
Well, it's better because it's louder, but there's still some noise. But maybe if you could do.
Can you hear me like now? Is it better like this?
It's more distant, but it's actually there's not this kind of mechanical noise on top of it. So I guess that's better. Sorry for interrupting.
So, okay, don't worry, it's fine. So mainly, what it's one of our main aim is also to investigate how the different types of traumatic events impact on the later on psychopathologies. This means, in our opinion, this means that experiencing, for example, abuse is completely different from experiencing a neglect during the childhood, and this should impact differently on the organism of the individual in terms of psychological outcomes and in terms also of biological changes. So here today I'm presenting the type of approach that we are using in this project. This project is funded by Sapienza University, and is a project that wants to investigate which changes, neurobiological changes, are induced by early life stress versus experiences, and how these changes are related with psychopathological risk later on. And the good things or the nice things about this project is that we use a multidimensional approach, because we really want to understand broadly the impact of the early life stress on the organism. As you can see from this picture here, we apply many different techniques to investigate such an impact. First of all, what we do is to profiling or characterizing the type of adverse childhood experience that an individual has in the past, and using different type of questionnaires, like, for example, the childhood trauma questionnaire, the parental bullying questionnaire. These are all questionnaires that usually investigate the type of traumas experienced and the type also, for example, parental care that each of us received in the first years of age. Then this individual is screened for several parameters, psychophysiological parameters. One is cardiovascular activity using the measurement of the heart rate variability. But also we investigated the level of the brain, the band activation, by using the EEG, the response of the brain to emotional pictures that characterize the activation of the brain. The other type of investigation we perform in parallel on the same individual is we observed the nonverbal behavior that an individual explains during an interview, a neutral interview. This means an interview that does not have emotional content. And finally, we have these other two characterization. One is the analysis of the blood biomarkers. So we collect blood from this individual, and we extract the RNA from there, and we measure the whole genome expression. And together in the parallel, we also collect saliva from the same subject, and there we measure telomere length, and we measure epigenetic changes, for example, global methylation. All these parameters are correlated, of course, with the psychopathological risk profiling. We use many different questionnaires that profiles usually depression, anxiety, or other type of symptoms. Today, what I'm presenting to you is just one part of these big studies that is going on, and we are now presenting the part on the heart rate variability. Just I want to mention why heart rate variability is so relevant, so important. So just to give you some idea, so the heart rate variability is an estimation of the variation between beats of your hearts. when a heart is healthy and is a good, usually the variability between beats is really high because it seems that your heart, your cardiovascular system is adaptable, is able to cope with the environment, with stress. So when an individual is, for example, in presence of stress, it can change the heartbeat and the frequency of the heartbeat because it's somehow adapting and coping with the stress. Instead, when a person has a not healthy heart, usually this variability is really low. And in the literature, it's been shown that this low heart rate variability is associated with depression, with the heart issue. And so it seems that the heart there is not responding, is not coping with the environment, the external environment with the stress. So keep in mind, low heart rate variability is something bad, high rate variability is something good. So those are the main things that you should remember. In this study, our aim was to investigate the impact of being exposed to a diverse environment in terms of parental environment. And here what we measured was that our idea was that the parental side, the type of care that we receive during the childhood impacts on the heart rate variability cardiovascular system. Our hypothesis was that an individual experiencing a negligent parental style is in a chronic physiological condition of defense that can be detected by this measurement by the heart rate variability. So in this study, we recruited something like 83 young adults. And for the psychological assessment, you use these two questionnaires. One was the BDI to measure depression. The other one was the STAI to measure the state anxiety and trait anxiety. And then we characterize the type of parental style each individual received during the childhood. And we use this parental binding instrument. Here you have an example of this questionnaire. You see the type of question that you can find in this questionnaire. For example, you have questions like that are related with the type of care that your mother or your father provides to you. As for example, the first question is, spoke to me in a warm and friendly voice. Or you have also a question that is investigating the control or the over control, the excessive control of the child. And there are questions like the third one, let me do those things I like doing. So in reading this questionnaire, you can have an idea which type of mother you have and which type of father you have, because you have one form for the mother and other one for the father. And in parallel, we measure the heart rate variability in a resting condition. So we can say that this is a trait measurement. So this is something that is really a characteristic of the individual. Interesting what we observe. Of course, we observe something that is known in literature, that having highly controlled parents, these are shown here by the gray bar, is something that's bad for the offspring psychology. Somehow you see that there is a significant increase of state anxiety in this group, and significant, together with a significant increase on trait anxiety, but also depression. So having low care, a high controlled parents is something that is affecting your psychology somehow, some of your behavior later on. But the most interesting things, in my opinion, is the impact of the parental care on the heart rate variability. Here we have results on the left obtained from the type of care the mother provides to the children, to the offspring, and on the right we have instead the father one. As you can see here, if you have an optimal parent in both cases, the case for the mother and for the father, there is a high level or a high heart rate variability. So your heart is beating with variability, so it's adapting to the environment. On the contrary, if you have either a mother or a father neglectful, this heart rate variability is decreased, significantly decreased. And the worst case is when you have a father that is controlling, so he provides a lot of control, and does not provide care. It seems that really having this father is impacting on these physiological measurements. This result is confirmed, these results are confirmed in population analysis, where you can see that here on the left that both maternal care and paternal care are significantly positively correlated with the heart rate variability. On the contrary, on the right, you have that the correlation between paternal control and maternal control with heart rate variability is significant only for fathers. So it seems that really the control of father on this parameter is the most significant, the most relevant one. Then I wanted to show you the results, preliminary results that we obtained from this another analysis that we are performing on the same sample. What is the results that we were obtaining by the analysis of the whole genome expression after collecting blood. So our idea is that when you are exposed to adverse childhood experiences, you have some biological alteration that mediates and somehow the psychological outcomes. And our interest is really to investigate which are the molecular pathways, the biological pathway that translate these experiences in psychopathology. Here what we did was to test 37 young adults, and these adults were classified by the type of trauma they experience, if they experience trauma, if they experience abuse, if they experience neglect, or if they experience different type of traumatic events, by using the same questionnaire that I showed you before, but also the CTQ questionnaire that is the childhood trauma questionnaire. And then we collect blood, and from the blood we extract PBMC and RNA. And we perform on this material a whole genome RNA sequencing analysis. We measure the whole genome in this case. So what I'm showing to you right now, what I can tell you is that these analyses are still ongoing, because this is the first picture that we got from this analysis, where that was obtained. This is the heat map that we obtained from this first analysis. As you can see here on the left, we have controls. On the right, we have traumatized subjects, and on each row we have biological pathways, and in each column instead we have subjects. As you can see here, it seems that there are some biological pathways that are differently expressed in controls compared to traumatized individuals. We are still analyzing the results, but at least it would be really interesting to understand. It would be really interesting to understand which are these pathways that characterize individuals after an experience, a traumatic experience in childhood. So just to summarize what we observed in this study, first of all, we found that experiencing low paternal care, a highly control, is impacting on the cardiovascular system and on this heart rate variability parameter that is really relevant. And specifically, it seems that having a father it seems that it's like this. It seems that there are data showing that having lower heart rate variability is something that makes you susceptible to cardiac vascular diseases. And also it's known that hereditary stress is connected with cardiovascular disease later on in the life. So that's quite it's known.
Okay, thank you. And the second question is, at what age is the difference in heart rate variability clear? Is there apparent change before and after traumatic events?
So here we have young adults. The mean age is 23 years of age. And it depends on the type. The parental side, something that you experience for a really long time, because it's from age of 0 to 18 years of age. So it's something that really lasts for a lot. It's really true that when we analyze these young adults, we are really close in somehow to this event. And not all of these guys, for example, show a psychopathological phenotypes in terms of psychological outcomes. It would be really interesting, for example, for us, longitudinally following these people in order to see if they develop, for example, later on psychopathology after this traumatic event, if this type of measurements that we have can predict this long-term susceptibility. But this depends if we get money in the next few years to do this longitudinal study.
Okay, thank you. I think in the for the time constraints, we should probably move on. Thierry, do you want to say anything else here?
I want to thank all the speakers and the questions for this session. And yes, I think it would be a good thing for the sake of time to actually give the floor to the next session. Thank you again, everyone, for their participation.